Just wanted to go over with you what we've talked about the last couple of days in class. We are starting this unit on cellular respiration, and then we'll do photosynthesis. Probably one of the more difficult subjects that we cover throughout the year. There's a lot of little processes that are going on in cellular respiration that are, they're pretty difficult. So I would encourage you to watch this video. Check out any other videos online about cellular respiration, hopefully help you understand it a little bit. So let's jump into this. So first of all, remember cellular respiration occurs in the mitochondria. It's going to occur in both plants and animals, because remember, plants do photosynthesis and cellular respiration. Animals just do cellular respiration. But this is a little picture that we looked at at the beginning of the year that shows, like in cellular respiration here, we take in glucose and oxygen. But remember, glucose can be any type of food that we eat. We're just representing it by glucose. We take in the glucose and oxygen, and we make energy for ourselves. And we give off carbon dioxide and water as kind of a byproduct that the plants use. But we're really wanting this chemical energy, and that chemical energy that we use is ATP, adenosine triphosphate. That's cellular energy. So all of the processes that you do on a daily basis, your body uses ATP to provide the energy for all those processes. Now, throughout the process of cellular respiration, there are a lot of oxidation reduction reactions. So there's a little trick to help you remember what those words mean. Oxidation, it's the mnemonic device oil rig. Oxidation is loss of electrons. Reduction is the gain of electrons. So for example, we can look at this chemical equation for cellular respiration, and what I tell my class is that oftentimes we can't see the electrons being moved, but you can usually trace those electrons through hydrogens. Okay? And so if we say oxidation is loss of electrons, if you look, the glucose is oxidized to become CO2. And you'll notice the hydrogens kind of go away in making of the carbon dioxide molecules. Reduction is gain of electrons, so oxygen is going to be reduced to water, H2O. And you can see the hydrogens being added there. So throughout the processes, this whole process, we'll look at some things that are being oxidized and reduced and so forth. Let's see. The next thing we want to talk about is the three main steps of cellular respiration. Glycolysis, the first one, which is going to occur in the cytoplasm. Also, one thing about glycolysis I'd like to mention is that oxygen does not have to be present for it to occur. It can occur with oxygen, but it can occur without oxygen as well. The second step is the Krebs Cycle, or also known as the citric acid cycle. And then the final step is the electron transport chain. So one thing I want to mention real quick too about cellular respiration is a very complex process. We are not going to take it at the extreme complexity of it. I figure if you're taking a college biology class, you could do that. But we're still going to go into it in a little bit of detail, and enough to where it will be a challenge for you, I'm sure. So let's take a look at glycolysis here. So one thing I want you to notice about glycolysis, we're starting off with glucose. This is a 6-carbon glucose molecule. It has 6 carbons in it. It's represented by these little green spheres. So the first part of glucose is we're going to split that glucose molecule. This is what we call the energy investment phase. So I like to use the old saying, you have to spend money to make money. And that's kind of what's going on with glycolysis. We have to spend ATP to make ATP. So right off the bat, you'll notice we're going to use 2 ATPs. And this ADP is adenosine diphosphate. So what we do is we strip phosphate groups off of ATP, and we add them to either side of this glucose molecule. So that now becomes ADP, which is a lower form of energy of adenosine diphosphate as compared to triphosphate. So those two molecules are going to be split by a series of reactions using different enzymes, which we're not going to go into. And then we come down into the energy payoff stage, and you'll notice here we're going to add a couple of what we call inorganic phosphate groups to either side of these 3 carbon molecules. We are going to then generate 2 ATP per 3 carbon molecules. So 2 ATP here and 2 ATP here. You'll notice this here and this here is exactly the same process is going on for both of these. Now the other you'll end up with a 3-carbon pyruvate on both of these, and that's kind of an end product. One other thing I want to mention is you'll notice this NADH. NADH is an electron carrier. Okay, so it's going to carry electrons to the electron transport chain. NAD plus is a molecule that picks up the electron. So if it has a positive charge and it picks up an electron, it's then going to be neutral. So NADH is this neutral that has picked up an electron. And remember what I said, we can usually trace those electrons by following hydrogen. So when it picked up that hydrogen there, that hydrogen ion, it also picked up an electron, and it now has a neutral charge. And it's going to carry those electrons to the electron transport chain. Really the main thing you want to gather out of this is the end result. So what I like to say, we used 2 ATP and we made 4 per glucose molecule for a net production of 2. Okay, so if there's a test question that says how many ATP were made during glycolysis, you make 4. Okay, but if the question says what's the net product of ATP of glycolysis, and it's 2. We're also going to make 2 pyruvate. Don't worry about the water here, it's really irrelevant to what we're learning about. And 2 NADHs, which again are those electron carriers that are going to go to the ETC, electron transport chain. It's important to note that most of the energy found in glucose remains in the 2 pyruvate molecules. That's going to take us to the next step, which is the Krebs cycle. Now there's a little kind of mini step that occurs between glycolysis and the Krebs cycle, and that's where pyruvate is converted to acetyl-CoA. Okay. The only thing that I really would care much that you knew about is that there is a carbon dioxide molecule given off in that step. It's the first time we've seen any CO2 given off. So there is a little bit of CO2 given off in that little in-between step. It doesn't even really have a name other than kind of pyruvate oxidation, I think is what it's called. But you'll notice in the Krebs cycle, it's a cycle because we're going to end with what we start with. So this will get added to a molecule and brought through the cycle, and it will end up with it and start the whole cycle again. I'm not going to go, there are a bunch of little intermediates along the way, a bunch of enzymes involved that we're not going to worry about learning about. The main thing that I want you to focus on, you to notice, is that we're going to give off a couple of carbon dioxide molecules. This is per pyruvate molecule, so per 3-carbon molecule, not glucose. Some NADHs, again, which are those electron carriers. We're going to generate 1 ATP per pyruvate or 2 per glucose. And then the last thing you'll notice we generate is an FADH2. That's a new molecule you probably haven't seen yet. That is really very similar to NADH. They are both electron carriers that are going to take electrons to the electron transport chain. So really the net result of the pyruvate oxidation and Krebs cycle is this 6 CO2, 2 ATP, 8 NADHs, and 2 FADH2s. So that's kind of the big picture that we're looking at with that. And then, so those NADH and FADH2 and the 2 NADHs from glycolysis are going to enter the final stage of cellular respiration, which is the electron transport chain. In this stage, electrons carried by those electron carriers from glycolysis and the Krebs cycle enter the chain, which is basically a series of proteins that pass electrons from one to the next, with oxygen being the final electron acceptor. So oxygen has to be present for this process to take place. Now here is what the electron transport chain looks like. I know when you first look at it, there's a lot of information on this screen here, but I'm going to try to break it down in the next few minutes to try to explain it to you. So what you'll notice here is you have these membranes. This is the outer membrane of the mitochondria. This is the inner membrane that kind of surrounds the matrix, and this is called the inner membrane space, and this is the matrix. So you'll notice right here, the little blue spheres, these are those proteins, okay? They don't actually look like this. This is just a model to represent them. Another thing I want you to notice is that here's our friend NADH and FADH2. They are going to be ones that are bringing those electrons to this electron transport chain. Notice the FADH2 will drop electrons off here on that second protein. It drops them off a little bit further down the line, so it can't provide quite as much energy. That kind of leads me to the next talk. So what the really important thing to see is that you see these little hydrogen ions, these H pluses. There's a lot of them here in the intermembrane space and not very many of them here. So what happens is these are going from this area are going to get pumped from a low concentration to a high concentration. Remember, that is active transport. Active transport requires energy to pump from low to high. So the energy that's provided for that comes from the electrons. So these electrons get passed from protein to protein to protein to protein. They pump the hydrogens into this intermembrane space, creating this hydrogen ion gradient. One thing I want to mention real quick, if we talk about the oxidation reduction, this NADH is going to be oxidized right off the bat. It's going to lose an electron. This protein will be reduced. Then this protein is oxidized, and this one is reduced. And this one is oxidized, and this one's reduced. So it continues that whole process of oxidation reduction, gaining electrons, losing electrons, gaining electrons, losing electrons. As we go down to the final electron acceptor is oxygen, right here. So the oxygen is going to pick up electrons, a couple of hydrogen ions, and form water. So that water that you give off when you breathe, when you sweat, when you cry, all that, when you urinate, that is the water that is produced through cellular respiration, more specifically the electron transport chain. So finally, once we have pumped all these hydrogen ions out here, they will then just follow their gradient through this last protein called ATP synthase. And ATP synthase kind of acts like a water wheel or like a turbine of a big water hydroelectric plant. These hydrogen ions flow through this and they generate ATP, large amounts of ATP. In fact, the largest amount by far. There's varying reports. Some books will say 32 or 34 ATP per glucose molecule. So this is again a quick summary. Hydrogen ions are pumped from the matrix by the movement of these electrons, I should add to that, into the intermembrane space, and then they flow through the ATP synthase generating ATP from ADP, and they approximately generate 32 ATP per glucose molecule. So that was really the first 3 steps of cellular respiration I wanted to go over with you. I'll make another video about fermentation, and it'll be a real short one. I'll probably make it sometime later on this week. Either today or tomorrow I'll be posting a little worksheet. It's not too hard, like a little study guide over cellular respiration to hopefully help you understand it better. It is a pretty complex process. I went through it very quickly. You might want to go back and watch it again. But anyway, let me know if you have any questions, and if not, I'll see you guys later. Bye.